Now that you know all about the first step of protein synthesis, transcription, in order to go on to translation, I need you to understand one key thing, and so this will be a very short video, but I need you to understand how to use the genetic code. When we get into class to talk about translation, it'll come very easily to you. So for this lecture, I'm going to talk all about what we call the genetic code. The genetic code is the way that we read, or eventually, let's throw this in there, translate the mRNA strand that you just made in transcription. It's the way that we read it, the way that we translate it. Just like if you were studying French or German or Spanish, a lot of times you have to translate into English when you're just learning so that you know what something says. So first thing and first big vocab word that I need you to know is codons. If I gave you this mRNA strand, AUG, GGC, UCC, AUC, and I could keep going. The way this is read first is we have to divide it out into what we call codons. A codon, if you've never heard this before, is a set of 3 bases. So whenever I ask you to take an mRNA strand and to turn it into a protein or an amino acid sequence, the first thing you have to do is take your mRNA strand, which is what this is, and you want to divide it out into codons, and it's just a set of 3 bases. My strand is read 3 bases at a time. So I would go through, there's 1 codon, there's 2 codons, 3 codons, and altogether I have 4 codons. Easy as that. Nothing hard about this one. Then what you have to do is take the codons and you have to use the genetic code. So if you wouldn't mind, turn to page either 11 or 18 in your packet and you'll see 2 different versions of the exact same thing. My favorite one is on page 11 and it is the pinwheel. In this one, if you use the pinwheel, you start in the middle of the wheel and then you work your way out in order to translate the code into an amino acid sequence, which is then eventually going to build your protein. This is the one that I will be talking about, the one that's on page 11, the pinwheel, and it's the one that I would always give you for a test or anything like that if I provided you with a genetic code. The other one, you read up one side of it. You read up the left side. Your first base comes off the left, second base comes off the middle, and then the third base comes off the side. Pick which one you like and get familiar with that one, but I will warn you, I will almost invariably use the pinwheel because it's my favorite, and I think it's the easiest one to read. You got your pinwheel, you got your genetic code. Now you've got to start breaking this down. So if you look at your pinwheel, my very first codon is AUG. AUG, if you start in the middle at A, then go to the U that's in that second part of the wheel for that little pie piece, and then find the other letter, which is G. And that should correspond to something called methionine. Methionine is unique, and I will talk about this one in class, so I do have a little bit more to say about methionine, but for now, just make sure that you got that part. My second codon was GGC, so do the same thing. Find your G in the middle, then go to the next G within that corner slice, and then find the C, which for this one, it doesn't necessarily matter because anything over in that little section for GG and then whatever the last letter is, it's always going to correspond to glycine, which is one of your amino acids. The genetic wheel is all 20 amino acids that are possible within your body. We only have 20 amino acids, that's it. Keep going. If I had UCC, which was my third one, start in the middle, U, go to the C, go to the C, and just like the last one, they all correspond to an amino acid called serine. The last one that you had was AUC. Find your A, find your U, and C, and then that is a amino acid called isoleucine. Okay? So you just have to follow the order. So anytime I give you an mRNA strand, break it down into its codons, then use those codons to then translate your mRNA code, that information from the DNA, into your amino acid sequence, into the protein, into the polypeptide chain that you are creating. Just to leave you with something to do, if I gave you the following mRNA codes, can you please tell me what my amino acid would be that would correspond to it? And I will give you 4 different ones. Your first one, what if your mRNA code, your codon, was AAA? What if it, for your second one, what if it was GCG? For the third one, try GAU. And for the very last one, for number 4, try CAA. We will continue here and we will talk about translation and how that actually works when I see you in class.